We've seen in previous videos that people like Ben Luft and David Persing and Norman Latov and Donald Marks have all said that the outcomes to Lymerix vaccination were very similar to what chronic neurologic Lyme is. And let's remember from the vaccine trial that these criminals deliberately did not report this type of adverse events. You see that here from Louis Bull. He was told, "Oh, there's no thing for me to check off here that says that you can have a chronic fatigue like outcome to Lymerix vaccination." So they deliberately blew all those people off. So for the past 10 years or so, I've been trying to explain to people how the OspA vaccine, given what it is, could cause this chronic immunosuppression outcome. And I get it every day from people, what are you talking about? It's still spirochetes, and yes it is, but spirochetes become wimpy after a while. And yes, you have to know exactly what Lymerix is in order to find out why it would cause an immunosuppression outcome. So let's start with that. So here's a picture of 3-cis or triacyl lipoprotein or tripalmitoylcysteine taken from the European Journal of Biochemistry by Bcellger. We can look at here, again, reminding you from the other videos and the other reports that we've done, that the NIH themselves, the NINS, or the National Institute of Neurological Disorders and Strokes, Adriana Marquez and Roland Martin themselves found that TLR2/1 agonists shed from Borrelia are responsible for immunosuppression in the body, i.e. no antibodies, yet chronic inflammation in the brain. Now, it's curious to me why it's not curious to anybody with MD after their names that this Lyme vaccine came on and off the market really quickly, and that the vaccine came off the market because it caused the same disease as chronic Lyme, being an endotoxin, being a fungal toxin. Why is that not curious to people like ILADS, or the infectious, well, I don't know what they call it. I don't even know what it stands for anymore. International Lyme and Associated Diseases. Why is it not curious to them that the vaccine alone caused this disease? Well, people would then say, well, if Lymerix caused the same disease as chronic Lyme, what are you treating? Obviously not spirochetes or co-infections. So it's important to know who is besides us saying that triacylated lipoproteins are the opposite of vaccines. So let's take a look at what else is a TLR2/1 agonist and across the board troublesome and cause immunosuppression in humans. BB, fermentin, syphilis, Staph aureus. And go ahead and go on PubMed and use a search term that's appropriate, such as PAM3-cis and TLR2, and find out for yourself whether or not it's true that these type of antigens are well known to cause immunosuppression in humans. Let's now look at what Nicole Baumgart says. Suppression of long-lived humoral immunity following Borrelia burgdorferi infection. Borrelia-infected host generates only short-lived, structurally abnormal and non-functional germinal centers. These germinal centers fail to produce memory B cells and long-lived antibody-producing plasma cells, leaving the host susceptible to reinfection with BB. This inability to induce long-term immunity was not due to the nature of the Borrelia antigens, as even T-dependent antigens of Borrelia were unable to induce such responses. So go to PubMed and search for B-cell germination centers and transformation. Why? Paul DeRay. Clinical pathological correlations of Lyme disease. This is ITSA's journal. Immature B cells can also be seen in the spinal fluid. These cells can appear quite atypical, not unlike those of transformed or neoplastic lymphocytes. Again, in 1992, in the book edited by Steve Schutzer, called Lyme Disease, Immunological Approaches, he says, on occasion these atypical appearing large lymphocytes have been misinterpreted in biopsy by several laboratories as cells of a malignant lymphoma or leukemia. BB antigens then may stimulate growth of immature lymphocytic subsets in some target organs, like maybe the lymph nodes, as well as in the cerebral spinal fluid. Unusual bacterial infections do not produce such lymphocytic infiltrates in tissues. These immunoblastoid cells in BB infections at times resemble those found in Epstein-Barr virus infections. Does BB reactivate latent virus infections in tissues? Do some inocula harbor simultaneous infectious agents? Parentheses, exoded ticks can harbor rickettsia, Babesia macroti, Ehrlichia bacteria, in addition to BB. End parentheses, producing multi-agent infections in some hosts. So where are we? You've seen that Baumgarth says that Borrelia burgdorferi infections seems to screw up the Bcellgerminal centers, producing incompetent B cells, rendering you generally immunosuppressed, not just to Borrelia, but subsequent viral infections like influenza. And Paul DeRay found immature EBV transformed like looking lymphocytes, especially in the cerebral spinal fluid, of chronic Lyme victims. So what do we see going on here? We see the production of incompetent B cells. Baumgarth seems to find some pathology in the germinal centers. Other people, like the NIH, have found that Borrelia burgdorferi, OspA-ish, triacylated lipoprotein-ish antigens cause the suppression of toll-like receptor 2 expression. It kind of turns off that function. It also turns off the HLA molecule. That is, other people have found that exposure to these types of antigens cause the immune cells to stop producing antigen. That might be what's going on in these immature B cells. They've been the process by which antigen is then moved in along the cell to the outer surface of the cell in the HLA molecules is arrested due to the nature of these lipoproteins. Baumgarth says it's not due to the nature of these lipoproteins, so let's investigate further. So in addition to the Lymerix vaccine having failed because it's an immunosuppressive triacyl lipoprotein, there were 3 other attempts I'd like to note that I could find, there's probably more, in the literature showing in parallel other vaccine attempts with this type. The first one says the 19 kilodalton antigen and protective immunity in a murine model of tuberculosis. They conclude the 19 kilodalton protein has a detrimental effect on the efficacy of vaccination with live mycobacteria. So it doesn't help. In other words, it doesn't cause immunity. The second one says Mycobacterium tuberculosis in 19 kilodalton lipoprotein inhibits Mycobacterium smegmatitis-induced cytokine production by human macrophages in vitro. They conclude, thus, the immunosuppressive effect is dependent on glycosylated and acylated 19 kilodalton present in the phagosome containing the Mycobacterium. These results suggest that the diminished protection against challenge with Mycobacterium tuberculosis seen in mice vaccinated with M. smegmatitis expressing the 19 kilodalton lipoprotein is the result of reduced TNF-alpha and interleukin-12 production, possibly leading to a reduced induction of T-cell activation. That sounds a lot like what Nicole Baumgarth found. The third one says, the Mycobacterium tuberculosis recombinant 27 kilodalton lipoprotein induces a strong Th1 type immune response deleterious to protection. They conclude, this study indicates that the 27 kilodalton antigen has an adverse effect on the protection afforded by recognized vaccines. So what do they say? Attempts at similar lipoprotein vaccines are deleterious, have a deleterious effect on immunity, an adverse effect on the protection, blah, 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 the immunosuppressive effect, blah, 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 diminished protection, blah, 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 reduced T-cell activation, blah, blah, blah. In other words, they don't work. Lipoproteins are the opposite of vaccines. So you absolutely have to know what Lymerix is in order to find out how it causes the same disease that chronic Lyme is, which is the same disease that was left out of the case definition at the Dearborn Conference. They wanted to create this vaccine, and they knew it caused immunosuppression, and they knew it caused no antibodies. So they say, okay, well, let's just narrow the disease definition to just the people who have the arthritis haplotypes, the people who have a tendency to produce antibodies due to an innate allergy response.